good afternoon, everyone. So I'm going to possibly carry on the theme that Claire set off beautifully about how important our microbiome is. And I loved how she started off by saying how we all are superheroes. What I would like for you all to think about possibly is how our immune system might be the superhero for us here. So what I'm going to try and talk to you about today is how our microbiome impacts our immune system. But firstly, why is this of interest for us? It is an aging conference, and I'm aware that you have seen this multiple times already. But what I do want to highlight here is that this big gap that we are currently seeing between our lifespan and health span is driving a major burden on our healthcare services. And there is a key drive now towards this concept of adding years to life. And what I want to say is possibly targeting our immune system might be the way forward. So what might be some of the key reasons why we see this big gap between lifespan and health span? And if I put my hat as an immunologist on, we can think about possibly we start seeing more infections as we age. So it doesn't matter whether they're bacterial or viral infections, we see more infections. And the bigger problem is that we also start seeing more hospital admissions and mortality due to these infections. And this was particularly highlighted well in the pandemic as well, where we saw the highest mortality in our older adults. So one of the key strategies that we have to combat this increased risk of infections is our vaccinations. But I've got further bad news that as we age, even the ability of our immune system to mount a robust response to these vaccinations is impaired. In addition to that, we also start seeing emergence of inflammatory conditions such as certain autoimmune conditions like rheumatoid arthritis. And what we believe is that all of these features are due to a remodeling of our immune system called immunesenescence. And what's emerging recently is that these features of immunesenescence could also be driving biological aging. So we have this beautiful paper out from Maria Miltenbrand's group that showed that if we aged your immune system in a young mice, we could actually drive biological agings of non-lymphoid organs such as your skeletal muscle or cardiovascular system. So the key take-home message is maintaining a healthy immune system as we age is of key importance. So what happens to our immune system as we get older? So because our immune system is responsible for fighting off a range of different pathogens, we have a number of different immune systems to do this. And what I'm going to particularly focus on today's talk is my favorite immune cell, the T cell. So I want you all to think of this one as commanders of our immune system. They are very important in fighting off viral infections, but they're also important in helping other immune cells to perform their functions as well. And unlike other immune cells, which go into our bone marrows to develop, your T cells, they migrate to this little lymphoid organ on the top of your heart called the thymus. And this is where they need to go to develop, maturate, and become functional. So firstly, what happens to our thymus as we age? So what we have over here is the thymus of a young person. And as we age, we start seeing a shrinkage of this thymus. Not only do we see a shrinkage, we also start seeing internal disorganization, architectural changes. So one of the key features is we start seeing an increased adipocyte infiltration. And what this results is in the loss of the functional niches where our T cells can go to develop. In addition to this, we also start seeing altered microenvironment within our thymus. So we see a loss of the thymostimulatory factors that promote this T cell development. And what's the result of all of this? Why are we so interested in thymic involution? So if you look at the young person over here, they have a lot of these naive T cells, or these are new T cells that have never seen a pathogen before exiting the thymus. And these are really important in helping us fight new infections. So what happens as we age, we start, due to this thymic involution, we start seeing fewer of these new or naive T cells exiting our thymus. One of the other key features of our adaptive immune system is something called memory. So if we break it down, during an infection response, we have our new T cells when they encounter a pathogen. So for T cells, it could be possibly a viral infection. They usually expand. But post the clearance of an infection, we have a small subset of these cells left behind called memory T cells. And the key function of this is that in case we re-encounter the pathogen, we have this pool of memory T cells to help us generate a robust immune response. So you might think here that that's fine. As we age, we have fewer naive T cells, so that means we can't deal well with new infections. But what we do have, on the contrary, is this expanded pool of memory T cells. So what do we think? If we re-encounter a pathogen in these older adults, would they be able to generate a good response? Now, I've got further bad news that although we might have more memory T cells, they are not necessarily functional. So like we have the hallmarks of aging, we've got hallmarks of T cell aging as well. And they're pretty similar features. So in addition to this shift in your subset distribution, we also start seeing features. For example, mitochondrial dysfunction is a very common feature. And the other thing we see is this buildup of senescent T cells. And these are particularly in your memory pool. So what happens is, although you have a lot more of these memory T cells, they have lost their ability to proliferate or expand when they re-encounter a pathogen. The other feature of senescent cells are that they are pro-inflammatory in nature. So they secrete, with the SAS phenotype, they're secreting a lot of pro-inflammatory features, and they contribute towards the inflammaging as well. So the key clinical consequences of these are that we start seeing a state of immunodeficiency. So there's a decline in ability to respond to pathogens, and also this inflammaging, which is a contributor towards other organisms aging as well. So what we believe is that the key success for healthy aging, so if you all want to be centenarians over there, is maintaining a healthy immune system. And my lab is particularly focused on looking at lifestyle-based interventions. So we've done a lot of work in the space of physical activity and how we can use that to boost our thymic output. We're also testing a number of pharmacological agents as well. But what I'm going to focus on today is talking about how we've been recently interested in the microbiome, particularly in this crosstalk between the microbiome and immune system, and how we can use microbiome-based interventions for healthy immune aging. So we've already heard a lot about how your microbiome composition changes with age, how we start seeing a loss of commensal species, but I want to introduce another concept called intestinal barrier dysfunction, or you might have heard about gut leakage. So this is very commonly seen in chronic inflammatory conditions, particularly those of the bowel. And what happens with age as well is that we have this intestinal barrier, which is your physical barrier, and it really helps keep your microbiome and even its products to avoid entering circulation. So it plays a very important role over here. But as we age, what we start seeing is that the appearance of gaps between these endothelial cells. And as a result, we start seeing translocation of microbiome, their products, and their toxins into circulation. And one of the hypotheses that we have is, so we've got some emerging evidence suggesting that this microbial translocation is a driver of inflammation. But what we were interested in exploring was that could this also be a driver of T cell aging? So to test this hypothesis, we started off with a human study in which we looked at healthy young, healthy older adults. Just to give an idea, so these healthy older adults that we have included, we had a very strict inclusion criteria. They're all healthy, no underlying conditions, no hospital admissions in the past 6 months. They have not had any antibiotics in the past 6 months, no therapies that could affect their microbiome. And we ended up with a really healthy cohort. And then what we wanted to look at is the serum levels of these microbial products that I was talking about earlier on. We've also, there have been a number of studies in mice as well as monkeys that have shown increased circulatory products or increased gut leakage. And the way we can measure this is we can look for occludin, which is one of your tight junction proteins. And if it's in circulation, that is a surrogate marker for increased permeability. And what we found here was that in the older adults, it wasn't any surprise that they had higher circulating levels of these occludin. But what we were particularly surprised about here was actually they were almost like we had two cohorts in there. One that was showing levels that were very similar to young people and other older adults who had high levels. So what we've gone on to do is then subgroup our cohort of older adults into those that show low microbial translocation versus those showing high microbial translocation, with the aim to see that if we could see differences in features of immune aging between these two cohorts. So we've done a number of phenotypic characterization and gene expression analysis. We've also looked at the fecal microbiota composition in these profiles, but I'm not going to go into that data today. So firstly, when we looked at your naive T cells, so these are the ones that have just left your thymus, I told you that the numbers of these cells declines as we age. But what we found was that there wasn't a significant decline between the levels of your young individuals and those older adults with low microbial translocation. We only saw this loss of naive T cells in this cohort of individuals, older adults with high microbial translocation, hinting that there is some link between your thymic output and these features of gut leakage. So on the contrary, if we look at your memory T cells, what we see is that we see this built-up of effector memory. So these are terminally differentiated memory cells which are not very functional and show a lot of features of senescence. And again, this was only seen in this cohort of older adults with high microbial translocation. So what we've gone on to then look at is your status of activation. So one hypothesis we had was that due to this translocation of microbial products, it was inducing this state of prolonged activation in our T cells. And what you can see here is that only those individuals with high microbial translocation were showing high expression of these markers of activation. And one hypothesis we had was that this prolonged antigenic stimulation and activation could be driving senescence. And again, what we found was that this buildup of senescent T cells, again, was only in this cohort of older adults with high microbial translocation. So I've thrown out a number of different immune aging features at you. And what I'm going to now introduce is, so we've heard about biological aging, chronological age versus epigenetic age, and we have another one in the block, which is called immunological age. So what we can do is, and this was done in support with our collaborators in Israel, is they've generated a metric where we could combine 12 different immune features that we can look at and generate one score, which is called your immunological score, which could be anywhere between 0 and 1. And the higher your immunological score, the more aged your immune system. So it's no surprise that older adults have a higher immunological score, but what we found was that there was a positive association between the circulating levels of these microbial products and your immunological score only in the older adults. So when we look at the features of your hallmarks of T cell aging, again, a very similar story. And I'd like to draw your attention here to a few genes. So for example, we heard about how important your autophagy processes are. And what we found was that this loss of autophagy-related genes only occurred in our cohort of individuals with high microbial translocation. Similarly, we also saw features of buildup of markers of senescence, again, only in this cohort. So far, I've shown you a lot of data showing that there's definitely a link between your gut leakage and T cell aging. But what we wanted to do possibly is go beyond just looking at this association and determine whether there's a causal relationship or not. And to do so, we have used a mice model. But before I get into that, the other thing I wanted to mention here is work that we've just started, so it's still initial days here. I told you earlier on that we were looking at really healthy older adults, but now we have moved on to looking at individuals who are possibly frail. So you heard about how frail individuals have a more state of this microbiome dysbiosis. And previous work from our group has shown that frail older adults also are down this path of further acceleration of immune aging. So any of the features that I've discussed earlier on, they are further enhanced in this cohort. So what we have done here is we were interested in seeing whether they also show even higher levels of this gut leakage. And to do so, we have looked at a cohort of hip fracture patients. So these are those individuals, and we've looked at them 6 weeks post-surgery. And the hypothesis behind this was that we didn't want to see any effects of surgery or hospitalization, but looking at the long-term effects. These were, again, individuals who had no previous underlying conditions or any comorbidities. And what you can see here is that in addition to this age-associated increase in our gut leakage markers, these levels were even higher in our frail older adults. And what we have just started to do now is look for links between these gut leakage features in our frail older adults and immunesenescence features. But as I said, moving on, we were interested in seeing whether there's actually a causal relationship between these two features. And to do this, we have used a mice model. So we've used a mice germ-free model. And when you age a germ-free mice, so these are mice that are raised in conditions, they have no microbiome. They are raised in a sterile environment. And a previous study has shown that these mice are protected from inflammation, increase as they get older, and they're also protected from this gut leakage that we were talking about. So we have looked at this occludin expression. For example, with the aged one, you see a loss, and our aged germ-free mice are protected from this intestinal permeability. So firstly, what we wanted to do is see whether they are also protected from thymic involution or not. But one of the first experiments we did was we wanted to confirm that do you even see a translocation of these microbial products into your into your lymphoid organs. So there's been some evidence suggesting that your microbial products can actually migrate to the thymus. And what we have seen is that your aged germ-free mice show significantly lower levels of these translocation of bacterial products because they don't show these intestinal barrier dysfunction. And if we further look into their thymus, so I told you with age, we start seeing a thymic shrinkage, and we also start seeing remodeling. We see increased adiposity, and that can be seen over here. So if we compare a young versus an old mouse, you can see more of these red stainings, and these are lipid droplets. And if we compare it to an aged germ-free mice, they don't show these features of buildup of adiposity. So they are protected from this adipose infiltration. The other thing we have looked at to see this maintained thymic architecture is over here we've looked at some stainings of lamin. So lamin is another feature of its membrane or your nuclear membrane protein, and the loss of lamin is considered as a marker for senescence. So if you look at a young thymus section over here, what you can see is densely packed with a number of thymic epithelial cells, lots of red expressing cells, so red is your lamin. If we compare this to an old mice, you can start seeing a loss of architecture. You have more of these black voids, which are possibly your adipocytes. And you also see a reduced expression of lamin because more of these thymic epithelial cells are now senescent. But if we compare this to your aged germ-free mice that were protected from the gut leakage features, their profile looks very similar to a young one's, where they show maintained architecture and fewer senescent thymic epithelial cells as well. In addition to this, we have also looked at the expression of both corticulary and medullary thymic epithelial cells, and it's exactly a similar profile. So if you look at a young mouse here, their regions are well defined. This is the medullary region in the middle and the cortex, and you can see a nice expression of the epithelial cells compared to an old one where they have fewer of these epithelial cells. And again, the architecture of an aged germ-free mice is very similar to a young wild-type mice. So in the first part of the talk, what I'm trying to suggest over here is a new hypothesis that this age-associated microbiome dysbiosis could be possibly driving your intestinal barrier leakage. And we can start seeing a translocation of these microbial products into your thymus. And this might be a new feature that's driving thymic involution because this could possibly promote features of senescence. We have also looked at inflammatory markers that were upregulated. And this could be in turn contributing towards this reduced output of your T cells by your thymus and overall contributing towards accelerated aging. We've also identified a number of bacterial species and metabolites produced by these bacteria as well, which are of interest and have been linked with this intestinal barrier leakage. So what I would like to focus for the last few minutes is now convincing you all that the reason behind all of this is that we want to use some microbiome-based therapies to target anti-immune senescence that have anti-immune senescence effects. And one of the first evidence that we had was using FMT studies in C. difficile mice. There's a lot of evidence on how FMT has a lot of clinical efficacy. So it can help combat inflammation, it can restore clinical symptoms in a number of inflammatory conditions. And what we've shown here is C. difficile infections, for example. There is also evidence that it can restore your microbiome composition and this barrier integrity. But what we have done is we looked at a cohort. So this was a small study in which we did longitudinal assessments of a group that was receiving. So these were recurrent C. difficile infection patients that were receiving an FMT. And we were interested in looking at the immune system to see can this restoration of your microbiome have potential anti-immune senescent effects. So it was no surprise that post FMT, so we saw an improvement in the microbiome homeostasis, and this lasted up to 4 weeks post FMT, where we followed on our patients. We saw a decline in your intestinal permeability, and we've looked at a range of immune features. So it wasn't a surprise that we saw an expansion of, for example, your anti-inflammatory immune cells. So these are the ones that secrete anti-inflammatory cytokines. We also saw a downregulation of the pro-inflammatory cytokine producing T cells as well. But what was surprising for us was that we saw post-FMC, so this is looking at a longitudinal analysis between one patient that responded to the FMT versus a non-responder. So if you look at the responder one, you can see that the final FMT, so they received up to 2 to 3 cycles, you see this fewer frequency of circulating senescent cells, which wasn't the case for the non-responders. So this is one of the first evidence that we had that your microbiome restoration has potential xenotherapeutic properties that we have heard a little bit about earlier on as well. And what we are particularly focused at the moment is understanding how and what features might be contributing towards this. So when we completed this study, one of our aims was that we wanted to actually test the beneficial effects on the immune system of FMT in a healthy older adult cohort. And we've had real challenges there. So one of the key drawbacks of FMT is this big donor variability that we spoke about a little bit. We received these samples. So at Birmingham, we are one of the few sites here in the UK that are licensed to do FMT. But we also receive samples from our collaborators in Canada and all over the world. And there's a big issue there between the donor variability and the comparability of the findings. So what we are trying to move towards is a more refined approach and a more precision-based approach where we understand which are the species and the metabolites that could actually be mediating these effects. So to do so, what we have done is we have set up our in vitro model in which we can age these T cells and induce senescence in them. And we are currently testing a number of bacterial products as well as species there to see if they have any of these xenomorphic properties. So if they could have anti-inflammatory effects and dampen down this SAS features. And we've identified a few of them that are of interest. One of them that I've shown over here, which is one of the metabolites that we are currently working on. So we are now even moved on to the next in vivo step where we're giving mice these metabolites to look at their effects. So firstly, what we did find was a dampening of inflammation and SAS features. But we've also shown that if we culture our T cells with this metabolite, you can reduce these P53 or senescence expressing cells. So you have a fewer buildup of senescent cells. And to develop an understanding on how this might be happening, we have looked at features of DNA damage. So another marker of your senescence. And what you can see over here is that your cells that were cultured with this microbiome-derived metabolite had a reduced buildup of DNA damage. And also looking at your mitochondria. So I told you one of the other hallmarks of your T cell aging is we see a build-up of dysfunctional mitochondria. And what we have shown is that if we culture our T cells with this metabolite, they have reduced mitochondria tracker expression. So that is looking at your total mitochondria mass. But we have now also confirmed that it has lower mitochondrial ROS production. And so it might be possibly triggering some of the mitophagy processes that we have heard earlier on about. So lastly, the other thing I want to touch upon is how we can use our diet as well to not only modulate our microbiome composition, but this could in turn also restore immune homeostasis. So in addition to collecting all the stool sample, we also collected a lot of diet data from our patients, from our participants, and via some integrative analysis, we have tried to identify features that associated with lower intestinal leakage as well as increased immune responses. So it wasn't surprising so we've heard a lot about the Mediterranean diet and its benefits. I never know it could even help with your fertility, so that was something new I learned today. But the other thing it can possibly help is to maintain immune homeostasis. What we found in our older adult cohort was that a higher adherence to this Mediterranean diet means they had a lower immunological age. Then we've also looked at these complex interactions between a number of immune features and these dietary components. But the key one that came up again was your dietary fibers. So we saw a significant association between your dietary fiber intake and levels of intestinal leakage as well. So again, what we are trying to propose here is that the prebiotics that we possibly heard earlier on about could, beyond just mediating beneficial effects on your microbiome, also restore your immune aging features. So I would like to end by sharing that how we have some positive news here. We have possibly suggested a few different routes via which we can restore your microbiome health, and that could in turn restore immune health as well. So for example, we heard a little bit about your probiotics, prebiotics, symbiotics, and add on another mix to that, which are called postbiotics. And these are actually delivery of the metabolites of interest. And the reason why this is of particular interest to us is that your immune system is actually capable of recognizing a lot of these metabolites. And that's how one of the key mechanisms of interaction between your gut, that the microbiomes in your gut, and your immune system in circulation. So we are currently working with a microbiome-based company to now generate a polybiotic supplement. So this is combining 8 species that we have identified of interest that could be potentially having immune-enhancing effects. The other ones are potentially fecal microbiota transplant. As I said, we have the postbiotics via your microbiome metabolites. We have dietary-based interventions such as a Mediterranean diet as well. So hopefully I want to end this at some positive news. That A, we want to maintain a healthy immune system if we want to live healthier, not only longer. And there are a number of different routes that we can achieve this while. So in the end, I want to thank the group, particularly my PhD student who just passed her Viva on Monday. So a lot of the work that I've shown is from her grad project, and my MRS student as well. And I'd like to thank all my funders, and a special mention to the BSRA. So a shameless plug, I'm one of the trustees of the BSRA, and we are hosting our annual scientific meeting at Birmingham next year. So hopefully you'll think about joining us again. And I'm happy to take any questions.
Time for a couple of questions. Gentlemen here.
There's been some speculation that actually the accumulation in the T cell compartment of memory cells might actually be driving thymic evolution, and which is the chicken, which is the egg. I was wondering if in your germ-free mice, if you had a look at whether or not they had larger numbers or less numbers of memory cells in general to the other mice.
So we don't see that many memory cells in your thymus, but what we are currently looking at in the germ-free model is the spleen, and to look at the memory built up there. But what we need to go beyond is we've looked at the numbers, but what we're currently doing is we're using the spatial transcriptomics method, which will enable us to see not only the numbers, but even look at their profiling. So we are currently looking at whether they have more, for example, built up of DNA damage or other markers of senescence as well, to give us some idea about functionality, which is also very important, going beyond just the numbers. But it's something we're currently investigating, so maybe next time.
Thank you. There was another hand, maybe, or no? Any more? Yeah.
I was wondering, in your patient cohorts, so healthy aged population, no like antibiotics used in the last 6 months, etc. What about like, standard medication that is typically easily used as people getting old? Even that proton pump inhibitor stuff that was mentioned before, I'm guessing a lot of these older people are probably on omeprazole, for example. How does it affect them or distinguish them from being?
So because it was a small study with only 55, we have excluded them if there were on any of those proton inhibitors, any statins, because we know statins can have immunomodulatory effects as well. Metformin, it's the other one. So these were all in our exclusion criteria. So one of the slight downside with this is we've ended up with possibly a biased population, which isn't a true representation of the aging population. And we also ended up with a very, so even if we looked at the diet scores, we've seen associations, but a lot of our healthy older adults were eating very well. So if we look at the 7-a-day intake, they were all almost achieving that, and that hints towards it as well, and hence I've shown the association. So we understand that we possibly ended up with a really biased cohort, but that's something we are investigating now. So we're doing two things. First, we're doing a longitudinal analysis. So this is almost 2 years ago that we started this study, and we're now carrying on yearly follow-ups as well to see how the dynamics change. But we've also, as I said, moved towards a more true representative population that has underlying conditions as well.
Thanks very much. I think we probably should move on actually. Thank you. Thanks very much, Dr. Duggal.